Today we are going to study Anthocerotae. As we are going to study Anthocerotae, let us study the classification. Classification of Bryophyta as proposed by G.M. Smith. G.M. Smith divided division Bryophyta into three classes, and these classes are Hepataceae, Anthocerotae, and Muscae. Hepataceae with 6 orders. You have last year studied the type Rixia that belongs to Hepataceae, class Hepataceae. So this year you have to study Anthocerotae. In that you have to study a type Anthoceros. So second class is Anthocerotae, which comprises of one order, Anthocerotales, two families, Anthocerotaceae and Notothylaceae. The third class is Muscae, which includes moss. And you are going to study Funaria from this Muscae. First and foremost, we'll talk about the distribution, where we can locate these. Members of this group are found in tropical, subtropical, and moist, warm, temperate regions. Mostly these grow on ground, some grow as epiphytes, some grow on the bark of the trees, and damp little extra. Moisture-loving plants. We continue with the general account. These are popularly called hornworts. Why? Because of the presence of the sporophyte, which is horn-like. The flattened green part is called the gametophyte. You can observe it here of the hornwort. Now let us see the external morphology of the gametophyte. How does it look like? It is small, yellowish green, dark green in color, dorsiventrally flattened and irregularly lobed. There is no distinct midrib. You can see here the second picture in which the sporophyte is quite visible. Dorsal surface varies from smooth to rough. On the ventral surface, there are many unicellular smooth-walled rhizoids. Mind it, in Rixia, Hepataceae, you have studied last year, the rhizoids are both smooth as well as tuberculated. Tuberculated rhizoids, scales and mucilage here are absent here. However, bluish-green spots of nostoc colonies can be seen on the ventral surface. Growth of the thallus takes place by a single apical cell or a group of marginal meristematic cells. Now let us study internal structure of the gametophyte. You'll have to see section through the thallus. The thallus shows simple organization. All cells are alike. Cells of epidermis are smaller. And photosynthetic and storage regions are not differentiated. As you see in case of Rixia, say it is the Rixia thallus structure of the Rixia. You can see here air chambers, air pores. You can see the two the halves of the thallus. The upper one, the photosynthetic region, the lower one, the assimilatory region. And air chambers and air pores are also absent here. You can see it is the Anthocerotae. Here you can see homogeneous mass of cells, and in between there are these patches like you can see here. These are the actually the mucilaginous cavities. Inside this are present the nostoc colonies, because it remains in association with, it is growing with an association with the nostoc, the blue-green algae. And here somewhere you will find the pores. These are called the slime pores. The slime which is being produced by the algae passes out through that. Now we are continuing with the internal structure of the gametophyte. Each cell of the thallus shows single, large, oval or flattened chloroplast. It has a large oval flattened chloroplast with a large pyranide in the center. Mind it, it's not a characteristic feature of land plants. Pyranoids you observing algae. Presence of pyranide is very uncommon in land plants. Now we talk about the reproduction in brief. It takes place by means of vegetative and sexual methods. Asexual reproduction is completely absent in Anthocerotae. Vegetative reproduction, it takes place by means of fragmentation. The older portions remain alive and they give rise to they get detached from the parent, and they can give rise to the new thallus. They reproduce by means of gemmi. Gemmi are small vegetative structures, outgrowth-like structures formed on the dorsal surface of the dorsiventrally flattened gametophytic thallus. These are vegetative reproductive bodies. They get detached from the parent, and each one gives rise to a new thallus. They also reproduce by means of tubers. Under unfavorable conditions, what happens? The thallus at different places forms these rounded structures. These are called tubers, and these tubers get liberated from the get detached from the main thallus, and they are formed under unfavorable conditions. Sometimes there is death and decay of the parent thallus, but the tubers remain there. They germinate to give rise to the new thallus. These Anthocerotae also reproduce by means of persistent growing apices. During dry season, if the thallus dries up, the apices at certain points remain alive. They remain persistent. They do not undergo any changes during dry season or unfavorable conditions. As soon as the favorable season comebacks, the persistent growing apices give rise to the new thalli. Apospory. Apospory means the thallus forms directly from the vegetative thallus, directly it forms. So it is called apospory. It is also observed in Anthocerotae. Sexual reproduction. How does it take place in Anthocerotae? Sex organs are deeply embedded in dorsal region of the thallus. The gametophytic thallus. Male sex organs are called antheridia and female sex organs are called archegonia. Genera can be monoecious or they can be dioecious, means both the male and female sex organ can be born on the same thallus or they can be born on the different thalli. Fertilization. Antheridia produce biflagellate male gametes called anthrazoids, while archegonia produce non-motile female gamete, the egg. The two fuse and give rise to a sporophyte. Actually, they fuse to give the egg nucleus and the anthrozoid nucleus fuse to give rise to a zygote. Zygote develops into a sporophyte. As a result of fertilization, diploid zygote or oospore is formed. It produces or develops into diploid zygote, which is the first cell of the sporophytic generation. It later develops into the sporophyte. Sporophytes usually grow from the dorsal surface of the thallus, each surrounded at its base by a tubular involucre and outgrowth from the thallus of the gametophyte, and it is protective in nature. The sporophyte is differentiated into foot and capsule. Seta is absent. Usually you see that the sporophyte gets differentiated into foot, seta and capsule, but here the seta is absent. It is green when young but turns grey or brown at maturity. And the sporophyte, children, you can see the horn-like structure. It is vertical in Anthoceros but horizontal in Notothylus. There is foot and capsule. There is no seta, but there is intercalary meristematic zone. And capsule is capable of unlimited growth in Anthoceros especially. Sporophyte is partial parasite on the gametophyte because it has also got chlorophyll present in its cells. As you see in case of the thallus, in the center lies the sterile tissue, which is called columella. It is made up of narrow, long, elongated cells. So it is a section through the sporophyte. You can see here the different, it's shown here. These are the different parts of the sporophyte at different levels of maturity. It gives support to the long, delicate capsule, the columella made up of sterile cells. Right in the center here, you can see in the picture. Inner to the wall is two-layered sporogenous tissue. You can see here inner to the wall. At the base, it shows archosporium, and here you can see archosporium, and later as you succeed, go towards the upper side. At successive higher levels, spore mother cells. Here you can see spore mother cells, then you can see spore tetrads, and here you can see spores as well as some pseudoelaters which are formed in Anthocerotae. Spores which are formed are thick-walled, tetrahedral in shape, vary in color from dark brown, black to yellow, and they are variously ornamented. The mature capsule dehisces from the apex, two halves. It just splits into two, twisting of valves, hygroscopic in nature, and they absorb moisture. Movement of pseudoelaters helps in liberation of spores. The spores which are formed, they are haploid. They germinate to give rise to the gametophyte. The gametophytic thallus will again produce the gametangia, the antheridia, and the archegonia, and again the fertilization will take place. As a result of fertilization, The male and female gamete will fuse to give rise to the zygote, and zygote will again undergo meiosis, producing the spores inside the sporophyte. You have seen how the spore mother cell is visible, and then at another stage you can see the spore mother cell undergoing meiosis, producing spores in tetrads, and then the tetrads getting separated, and you can see the spores, and as it matures it liberates the spores. Okay. Alternation of generations. Two clear morphologically distinct generations, gametophyte and sporophyte. The gametophyte is simple thallus, it is autotrophic, it can manufacture its own food material, and it bears the sporophyte because the sporophyte is partially parasitic on the gametophyte. It is only partially parasitic because it doesn't have rhizoids and all that, with which, by virtue of which it can just hold it. It has food which is embedded inside the gametophytic tissue, gametophytic thallus, and it has got nocita, and it has got meristematic intercalary region, and ultimately the capsule. You can observe the graphic life cycle of Anthoceros. And at the end, a big thank you to all my students for patiently listening to my lecture. Please like it, share it, and subscribe it. Thank you.